Good afternoon, students. Today we are going to start with Unit 3 of Industrial Microbiology, and that is Inoculum Development or Inoculum Buildup. So the first topic of Unit 3 is Inoculum Buildup or Inoculum Development. Now let us first understand what is inoculum. What is inoculum? Inoculum is the mixture of cultured microorganism. Inoculum is nothing but a medium containing actively growing microorganisms. Okay. So inoculum is the mixture of cultured microorganisms along with media in which it is growing, or you can say it is a broth culture. Okay. What is inoculum buildup? Inoculum buildup process is a stepwise and gradual increase in scaling up the volume of inoculum up to the desired level, which includes preparation of bacterial suspension, either vegetative cells or spores, in sterile tap water or nutrient medium, and then to the broth, or in case of fungi, their hyphae are transferred to the broth. Okay. So today we are going to discuss about inoculum buildup. See, inoculum is nothing but microorganisms cultured in media, or microorganisms along with media in which they are growing. Now, why it is necessary to study inoculum buildup? When we are carrying out any fermentation process, or when we are carrying out large scale production of any substance, then we need microorganisms to be inoculated into the raw material, or to be inoculated on the substrate. And we have the microorganism which will be inoculated. Then why it is necessary to study inoculum development? Because a large amount of inoculum needs to be added to a large fermentation tank. Suppose your tank is having a capacity of 30,000 to 40,000 gallons. Then you need to add minimum 0.5 to 5% of inoculum, and in certain cases 20% of inoculum of the capacity of the fermentation vessel. So you need a large quantity, large volume of inoculum. And a large volume of inoculum cannot be produced by using a loopful of culture. So, inoculum buildup is actually a gradual or a stepwise process that we are going to study. So, inoculum buildup is a stepwise and gradual increase in the volume of inoculum. Right. So, inoculum production is a very critical stage in any industrial fermentation process. If we inoculate a loopful of culture into 30,000 to 40,000 gallon tank, a prolonged incubation period would be required for visible growth of microorganism to occur, and also for product formation. You can just imagine, when you are doing experiments in your laboratory, then a loopful of culture to be transferred into a test tube or a flask or on a nutrient agar plate is sufficient. But if your tank is having a capacity of 30,000 or 40,000 gallons, and such large volume of nutrient medium is present, and if you inoculate a loopful of culture, then most of the time the microbes may die, or a very long incubation period will be required for visible growth to occur, and naturally a long period will be wasted for product formation. So inoculum buildup cannot be done by inoculating a loopful of culture into the large production tank. Thus inoculum buildup or inoculum is prepared in a stepwise sequence, employing increasing volume of medium. So say for example, first you have a stock culture, you inoculate a loopful on a slant and you get a slant culture. Then in the next flask you may be having 200 ml medium, then a 4 liter tank, then a 10 liter tank, then a 100 liter, then 1000 liter, and so on. That means you gradually go on increasing the volume of medium, and you go on inoculating at every level the microorganism, right? So approximately 0.5 to 5% inoculum by volume is transferred from previous step to the next step in the sequence, except the initial inoculum from a stock culture. When you have to take a loopful from stock culture, or if you have spores, then you have to follow a particular procedure. Otherwise, in each step, 0.5 to 5% inoculum is transferred from one stage into another stage. Okay, so if you look at the diagram, you can see that you have a stock culture. You inoculate a loopful into the maintenance culture or slant culture. Then you transfer the culture into, you scrap the growth and transfer it into a Erlenmeyer flask containing seed culture, say 200 ml. Then you transfer 5 to 0.5 to 5% of the broth culture from Erlenmeyer flask. Okay, so from Erlenmeyer flask 0.5 to 5% culture into the fermenter having a capacity of say 4 liter. Then you transfer into the next tank having a capacity of 10 to 10 to 40 gallon fermenter, and then to the production tank. Okay, and every step, how much of inoculum do you add? 0.5 to 5%. Except in production, sometimes 20% inoculum or more also may be required. So from the diagram you can see that you go on increasing the volume of medium, and hence you go on increasing the volume of inoculum or broth culture. And that is how why we say that inoculum buildup is a stepwise process. Okay. Next thing, why we need to do inoculum buildup? Organisms must grow quickly. The organisms must grow quickly and in enormous number at each step, so that incubation period required is very short, right? So organisms must go quickly in enormous number and incubation period should be short. So we need to follow inoculum buildup. Further, the inoculum medium is balanced in such a way that it will support rapid growth of cells, that means rapid growth of microorganism, and not product formation. Okay, usually 0.5 to 5% inoculum is introduced or added to the production tank, but sometimes we require as high as 20% or greater medium. When do we require high level of medium? When conditions such as presence of growth inhibitors or poor energy recovery of cells does not favor extensive growth of microbes, right? Matlab, when do we add 20% of inoculum or more? When certain growth inhibitors are present in the medium or recovery of cells does not favor extensive growth. Okay, say for example, if you are using bismuth, yeast, glycerol fermentation and all, in that case you have to add 20% or more of culture medium. Otherwise 0.5 to 5% inoculum is sufficient. Then the quality and reproducibility of the inoculum are also critical factors in getting or in reproducing high yield from one production run to another, and in obtaining valid experimental results in small tanks used in research studies. Okay, the quality of inoculum is important, reproducibility is important. When you want, when now there are critical factors like reproduction of high yield from one production run to another, especially when you are carrying out the studies at research level. Okay, so when you are carrying out studies at research level, that means when you are carrying out, you are studying a new production first in laboratory, then on a small scale fermenter, glass fermenter, and then on then in a pilot fermenter. Then if you are studying the production in lab, then in industry, then sufficient amount of inoculum. So for all these studies, enough inoculum should be available in a single batch, so that you can split it uniformly, so that each tank gets the similar inoculum. Okay, so each tank or each study should get the same inoculum, and that is why you should have sufficient enough quantity of inoculum. Further, many generations, okay, this point is also be taken care of, that many generation of microbial cells occur when you start the inoculum buildup and finish the inoculum buildup, or when you start inoculate from stock culture into the slant culture, and at the end you have a large tank filled with broth culture or inoculum. So many generation of microorganism occur. What we assume that during all these cellular multiplication cycles, the microorganisms do not change physiologically or genetically. We assume that the organisms don't undergo any genetic change or any physiological change. But this is not always the case. Microorganisms may undergo mutations. Though the frequency of mutation is low, microorganism can undergo genetic change or mutation. Now, see, these mutations are of no worry. We don't have to worry about these mutations if they are not causing any change in the organism, or most of the times the mutations are lethal. And if most of the times the organism do not cause, the mutation do not cause any positive change in the organism or some kind of change in the organism, it is not a problem. But mutations are a problem when the because of mutation the ability of organism to produce the fermentation product may change. Okay, if the ability of the organism to produce the fermentation product changes, or some added advantages are gained by the organism because of mutation, then it can be a problem. Okay, now if for the production you are using a mutant strain itself, that means if the organism used for the fermentation is itself a mutant strain, then it serious problem can exist. Why? Because mutations are not stable and you are using a mutated organism for the production, and mutations are not stable. So, the frequency of back mutation or loss of particular mutation is rather high. That means you are using a mutated organism, but during the course of fermentation, the organism may undergo back mutation and revert back to the original state, and the chances of this back mutation are high. So, whenever you are using a mutated strain, stock culture maintenance of mutated strain should be done very critically and carefully. You have to take care that the mutated organism does not revert back to the normal state or does not undergo back mutation. That is to be taken care of judiciously. Right. Now, when you are transferring microbial growth from stock culture to liquid medium, and initiation of growth in liquid media at times create problems as regards the initiation of inoculum buildup. What do we mean by initiation of inoculum buildup? That means you start the process of developing inoculum. What happens when you are starting the process of developing inoculum? What you do, you take some amount of culture or microorganism from the stock culture and start the inoculum buildup. That means you initiate the growth of the organism in liquid medium. But sometimes there is a problem, especially when you are using spores of microorganism like bacterial spores, fungal spores or actinomycetes. Then there are problem in starting the growth, initiating the growth in the liquid medium. Okay, so we face various problems. Okay, various problems are faced during initiating the growth of microorganism in a liquid medium when you start inoculum buildup. So how do we tackle these problems, or how these problems are to be taken care of? Okay, now vegetative cells or spores of bacteria are first suspended in a sterile diluent like tap water for addition to broth medium. Okay, what we have to do, vegetative cells or spores of bacteria first suspended in a sterile diluting agent like water, and then you add it into broth medium. But there are certain spores which require heat shock treatment to allow germination of high percentage of spores. The best example is highly heat resistant spores of Clostridium acetobutylicum, which is used for acetone butanol fermentation. Right. These spores do not germinate readily. They need to be given heat shock treatment. Okay, before inoculum buildup. So how do we do? How do we give heat shock treatment? The spore suspension or the spore culture of Clostridium acetobutylicum used for acetone butanol fermentation is placed for 90 seconds in hot water and then it is rapidly cooled. Okay. Okay. So when you heat the organism, initial organism to a very high temperature and then rapidly cool it. This is called as heating followed by rapid cooling. This is called as heat shock treatment. When you give heat shock treatment to the spores of Clostridium acetobutylicum, then only they germinate. Okay. So there are certain problems with initiation of growth of microorganism in the beginning of inoculum buildup, and that is to be taken care of. Okay. So this is how you have to perform heat shock treatment for spores of Clostridium acetobutylicum. So you take a corn mash, that means corn medium is there, and you inoculate it with or put the spores of Clostridium acetobutylicum, put it in a boiling water bath for 1 to 2 minutes. Okay? And then you have to immediately transfer the tube into ice bath. So you heat it to boiling and then you rapidly cool it. This causes heat shock. This gives organism a heat shock, and hence the spore coat loosens and the organism germinates. Right? The spores of fungi and actinomycetes do not require such heat shock treatment, but they require pre-germination in a special medium. Okay. Spores of actinomycetes and fungi do not need heat shock treatment. They are not so heat resistant, but pre-germination of these spores is to be done in a special medium. Okay. So spore suspension of fungi and actinomycetes are prepared by adding suitable diluent like sterile tap water to sporulated agar growth. Okay. So what you have to do is take a test tube containing sporulated medium. It's a agar slant containing spores of either actinomycetes or fungi. What you have to do? You pour some sterile diluent like sterile tap water in the sporulated agar and scrap the spores. Okay, with the help of a loop, inoculating loop, sterile loop, you scrapping to lose off the spores with the help of sterile loop, and you have spore suspension ready to be inoculated into the or ready for inoculum buildup. Okay, but there are certain fungi or certain organisms whose spores do not get wetted by sterile diluent like water. Okay, you add water to the medium, scrap, but the spores do not get wetted by the diluent like water. So in that case, they float on the surface of the medium. The spores float. Excuse me. This condition is corrected by adding some small amount of non-toxic sterile wetting agent like sodium lauryl sulfonate to the dilution medium. Is that clear? Spores of fungi and actinomycetes are supposed to be given pre-germination in a medium, and for that you take sterile diluent like sterile tap water, put it in the test tube of the spores, scrap the spores, and the spores will get wetted up or mixed up with the diluent, and then you can pour it in any inoculum medium. Okay, inoculum buildup medium. But there are certain organisms whose spores do not get wetted up by the diluting agent and they float on the surface of the liquid. In that case, we add a sterile non-toxic wetting agent. Okay, the wetting agents reduce the surface tension so that the spores get wetted up or they come down into the medium. What we can do this by adding sodium lauryl sulfonate. Okay. So this is how we can do. You have a fungal mat containing spores. Put sterile distilled water into it and scrap with a sterile loop and prepare a suspension. But in the second diagram, you can see that the fungal mat or the fungal spores are floating. They do not get wetted up by the liquid. So you add a wetting agent like sodium lauryl sulfonate so that the spores get suspended or dissolved in the sterile diluent, and then you can add it into the inoculum development medium. Okay. Now, in contrast, certain fungal fermentations, say for example Aspergillus niger, used for citric acid production, require that the fungal spores should float on the surface of the liquid medium. Okay. When we are carrying out citric acid production using Aspergillus niger, we use surface culture production method where the organism is present only on the surface of the medium. So what we do in this case, we want that the spores should float only on the surface of the medium. How can we do that? We can do that by spraying the spore suspension only on the surface of the medium by atomizer. Like a spray bottle, you have a spray bottle. So an atomizer bottle is like that of a spray bottle. So the spore suspension is atomized on the surface. It is sprayed on the surface of the liquid medium so that organism remain only on the surface and carry out citric acid production. Now, but at each and every step of inoculum buildup, there are chances, there is a risk of contamination. Okay, so risk of contamination is always present in inoculum buildup. Contamination can lower the production, final production, product yield can be reduced. It can kill the microorganism, or there can be competition with the contaminating microorganism. So at each and every step we have to take effort so that there is no contamination. You have to prevent contamination, and at each step you have to detect whether there is contamination or not. Contamination can be checked by sterilization methods, and it can be detected by offline online monitoring. Contamination is generally detected by microscopic observation. You can microscopically observe in between the medium. You can stain it and check whether the wanted organism is present and unwanted contaminating organism is not present. You can culture the sample on selective medias and check whether contamination is present or absent. When you are using anaerobic microorganism, then anaerobes are to be cultivated using special anaerobic condition and check whether contamination is present or not. So these are all the steps that need to be followed during inoculum buildup. Okay, so that is all with inoculum.